Hi everyone, it's Mrs. Shannon. I hope you guys are doing well. As you have heard by now, I am home with a broken foot and I will be getting surgery this Tuesday and hopefully will be out 2 weeks and can return. I really miss you guys and I wish I was there instead of here. But in the meantime, I do want to get back to what we started before I left. And I know that you guys have been doing an awesome job and have been really focused and concerned about what you are learning. And I want to go through the rest of our notes so that way we don't get behind a little bit because you do have our I Learn coming up. And I want to say it's going to be in April. And so I want to make sure that we are able to get through everything and you learn it well. Also, there has been concern about the last weekly review. You will have a chance to retake it. I posted in Power School a review packet. There's also a quizzes that will be very helpful. As soon as you take that and submit it, I'll go through later this week and I'll see who has. I'll get in touch with those people and set up a way for you to take a re-quiz. You might have one more weekly review while I'm out for this duration. Again, it will be open notes, so I just want to give you that heads up. When I return after this next unit that you will be starting on Tuesday, we will do a big exam on cell division, cell cycle, and this new stuff that we'll be learning on protein synthesis. But that will be down the road. You will have plenty of time to learn, ask questions, and I will be back for that, okay? So let's get back to business. I think this is where we left off in our cell division and cancer notes, or if it's called cell division and cell cycle, I'm not sure what I called it. But hopefully you have with you the strips that we were using as headings and you have your notebook open. So if you have those headings and strips, I believe we were ready for strip number 5, the cell division mitosis. We had talked about why cells divide. We have talked about chromosomes and the structure of chromosomes. We've talked about S phase and replication. And now we're going to get to the part of the cell cycle where the cell actually divides. Remember, cells divide, growth, and repair. In some organisms, some simple organisms, prokaryotes mainly, such as bacteria, mitosis can be a way for those single-celled organisms to actually reproduce. But since it's such a small amount of organisms, I guess I should say phylum that we're talking about, we're going to focus on the eukaryotes, okay? And so we're going to focus on the growth and repair as being the primary reasons that mitosis occurs. Just remember, yes, a couple of organisms can reproduce using mitosis, or not mitosis, a form of it called fission, but it's very similar to mitosis. Okay. So in mitosis, we start with one parent cell and we end up with 2 daughter cells. And I don't remember, we may have gone through the slide. I'm not quite sure. So if I have, you guys can just go through it again or forward to the next slides. But notice that the parent daughters, they are identical. We're making identical one. And this is important because later on we're going to talk about how eukaryotes reproduce sexually and how these daughter cells will not be identical to the parent. But for now, during mitosis, they are identical. One parent, 2 exact daughter. This occurs in all somatic cells. A somatic cell is any body cell that's not any body cell. The opposite, I should say, of somatic cells will be your reproductive cells, such as your sperm and your egg, also known as gametes. Okay, but if it's not sperm and egg, then we're talking somatic cells. So mitosis is a form of cell division when one cell becomes 2 identical daughter cells. Don't forget, I'm doing a screencast so I can talk about it, but also you can hit pause at any time. Many of you have done the Amoeba Sisters and have done other activities already to explore mitosis. Now, when we're looking at mitosis, we divide mitosis into 4 different steps. And when we're talking mitosis, we're focusing on the nuclear division. Right after the nucleus divides, then the rest of the cell divides in cytokinesis. This is where, depending on what source you're looking at, mitosis may or may not include cytokinesis. Sometimes mitosis just includes these 4 steps and focuses on nuclear division, and then cytokinesis is separate. So just kind of keep that in mind. But you will want to memorize PMAT. Okay, PMAT are the 4 distinct stages of mitosis. Oopsie, sorry that I had it written here. We have prophase, metaphase, anaphase, and telophase. Prophase is going to start, let me see if I can actually, nope, I cannot pause it, so I'm going to have to show you. Okay, right here, when we start to get together, we can see the chromosomes, that's prophase. The nuclear envelope starts to split. Metaphase means that we meet in the middle. Meta, meet. We meet in the middle. They both begin with M. Anaphase, we just went past, was when they start to get pulled away. So let's watch this one more time. Prophase, we can start to actually see the distinct chromosomes. They become tightly coiled so they don't get tangled up. Metaphase, meet in the middle. And then away, they go away to opposite sides in anaphase. And then telophase is when the 2 nuclear envelopes form and separate them. Okay, so pro means before. It's what happens before we actually divide. It's when the cells condense. Meta, M, think middle. They meet in the middle. A, away, anaphase. They go away from each other. And T for 2 new nuclei. Okay, and here we go. This is not an animation, but it breaks it down. Prophase, you can start to see the chromatin starts to coil and become more distinct into the chromosome shapes. The nuclear surrounding envelope, the nuclear membrane, disintegrates. In metaphase, they meet in the middle. Anaphase, they start to go away to either side. Telophase, 2 distinct nuclei start to form. And then cytokinesis. After this nuclei form, then the rest of the cytoplasm and cell organelles just kind of randomly separate and split up into the new cells. So you can remember cytokinesis is when the cytoplasm divides. Okay, so the next slip of paper or heading we had was, how often do cells divide? And it really depends on which type of cell it is. For example, if we have cells that are on the internal lining of the intestines, they actually divide about every 5 days. Skin cells, okay, they undergo mitosis every 2 weeks, and red blood cells every 4 months. And then you have some cells that once they become what they are, they don't even really divide at all unless something happens that they need to repair themselves. Liver cells, 1 year. And so you can see the different lengths of time. Now, why they divide, why do these body cells divide? Again, it's growth and repair. And I know I keep saying it over and over, but I need you to have that down. To grow bigger, an organism grows bigger this way, but it also repairs. Okay? So, cell cycle. It takes different amounts of time. They divide some cells more often than other cells. We've just learned that. We've learned the process of cell division called mitosis. We've learned the different parts of the cell cycle. And the next thing we need to talk about is regulation of the cycle. How do the cells know when to divide and when not to? How do they know, okay, it's a good time to divide, we should go ahead and do it. Oh, we need more of us, let's go ahead and do it. How do they know that? And so now we have regulation of the cell cycle. And this is a new tab as well, a new heading from your paper headings. So if you want to pause it and tape it in, now would be a good time. Okay. So the cell cycle is controlled by a chemical control system. There are chemical signals, chemical messengers that starts and stops events in the cell cycle. So we talked about these different phases like G1 and S and G2, and it's all controlled by chemicals in our body. Now there's an external regulation. These are signals that come from outside of the cell. Examples would be like nutrients in your body. Okay, if you need to produce certain cells to break down certain nutrients that are coming in, then you will need to produce, you will need to have signals that are sent to the cell to divide. If you have something toxic come into your body, okay, certain cells will need to take care of it, and you might, the cell might divide to make more of it. Hormones are really common chemical signals that are moving throughout the body that trigger cells to go to undergo undergo cell division. So cell signals can come from outside the cell, but they can also come from the nucleus. For example, the DNA inside the cell. There's instructions within the DNA. There are certain genes that code for how often the cell should undergo cell division. And we call these like signals and stuff, we call them messengers, chemical messengers, or chemical signals. In addition, there's checkpoints that the cell will undergo to make sure that it's ready to go on to the next part of the cell cycle. It's a critical point in the cell cycle where stop and go signals can either stop a cell from proceeding on in the cell cycle and eventually dividing, or it can signal that the cell should go ahead and proceed on to the next phase of the cell cycle in anticipation of cell division. Most of the time, cell division is off, unless there's some sort of signal or stimulus to create more. When you did the HHMI Click and Learn, this talked in depth about the signaling, the checkpoints. It focused on one called P53 and how it helped to proceed from one phase to the next. Now, if at any point a cell is not able to go on, it's something, there's a chemical message that signals that it is damaged, that there is a problem. It enters what's called apoptosis. Apoptosis is programmed cell death. It's either an internal or external signal that activates the genes that produce self-destructive enzymes. It tells the cell to make enzymes to break itself down. Okay? When this happens, the nucleus shrinks and breaks apart. Apoptosis prevents abnormal cell division and cell growth. It prevents damaged cells, mutated cells from going on. Sometimes this leads to visible traits that we can actually see. So like when we see web fingers and toes, if anyone has it, it's because apoptosis, this breakdown of the skin cell, was not completed here. There was an issue with the apoptosis. Cancer is uncontrolled cell division. Okay, the cell does not break down. It happens when the regulation of the cell cycle breaks down. They divide much more often than healthy cells do. It leads to the formation of tumors, which is a clump of cells that divide uncontrollably. And if this is damaged cells that divide uncontrollably, you're making many more cells that have the irregular DNA or the irregular formation of other parts of the cell. There are two types, malignant and benign. Malignant tumors are when cancer cells break away from the tumor and they are carried to other parts of the body and form more tumors. Okay. Benign tumors usually remain clustered. They're easier to remove as well. It may be harmless and easily removed. Okay. Causes of cancer often cancer can result from exposure to carcinogens, which are any cancer-causing agents. Tobacco smoke, UV rays, for instance, X-rays. What happens is these carcinogens mutate or change the bases in the DNA somehow. Other random mutations can happen from infectious agents such as HPV. Now, there's cancer cells are not necessarily caused, though, by exposure to carcinogens or other mutations. Sometimes there's a predisposition for cancer already within the gene itself, and it just takes one small trigger to activate it. There's a famous line of cells called HeLa cells that I want you guys to know about, and if you're ever interested in reading book pertaining to science, this is a great book to read. In fact, some high schools, I don't know if Concord's going to do it, their AP Lit class actually reads this. A HeLa cell is a cell type that is an immortal cell line used in scientific research. It is the oldest and most commonly used human cell line. What happened is there was a woman named Henrietta Lacks who had cervical cancer. They took her cells without her consent or her knowledge so that they could study them. And they actually used those cells to study cell division. A lot of research has happened, but there's a lot of controversy because of taking this without her knowledge, her permission, and her consent. And so if you want inside the actual screen or inside the actual slideshow, there is an actual article on Henrietta Lacks and HeLa cells that you should be able to access from the Smithsonian Magazine. It's taking a while here. And you can find out a little bit more about Henrietta Lacks. Finally, vocabulary that you should know. There's a list of vocabulary terms that have come up throughout the past couple of weeks. We did not spend much time on cell differentiation. I have a lab for cell differentiation. I would like to be there to talk a little bit about cell specialization and differentiation. So for right now, I'm going to kind of leave that off to the side. We will come back once I return to actually do the lab and talk about what it means for cells to differentiate and become specialized to do one job. And we'll see how one set of DNA, one set of blueprints can lead to all these different cells with all of their different jobs. So we will come back to that section. So you can kind of ignore the cell differentiation and cell specialization for now. Now, when you are done here, hopefully your notes are on the left side of your paper, and your right side of your paper is probably blank unless you've already started reading. At this point, if there are areas that you're confused about and you're going, Mrs. Shannon, I really don't understand what the heck you were saying, or I need more information, this is the time to go to CK12. So I actually tweaked the link, so now it's going to take you, so you don't have so many options to choose from, to just what we call Biology CJHS, Concord Junior High School, Flexbook. Right now, there's only 2 sections. I'll be adding more sections for the next unit that we do. And you can click on these to find information. Now, what I want you to do is not copy down every single thing that's in here. Just fill in the gaps. What did you still have questions about? What was not covered? Are you able to answer that topic statement or question and talk about it? Or do you need some more help? Are there vocabulary terms that you don't know about? If so, that's what you should be writing down on the right-hand side. These notes are for you. I am not grading these notes, meaning you don't need to worry about how much you put down. It's the type of notes that are going to help you that's important. Okay? So right now we have 2 sections for you. Finally, I added in another game. I know I put a lot of interactives in places. Last week you had quite a few, but there is a DNA replication game. If you were struggling with DNA replication, I added this in. I don't think I put this in there before. You might have to activate the Adobe Flash to do it, but what it's going to do is it's going to have you practice looking at the structure of DNA and then practicing base pairs and matchups. And so I would really suggest, if you have time in class, go ahead, play it, and see if that helps you understand replication a little bit more. So I hope these notes help. Tomorrow, you guys will be starting a unit that is more self-guided, where you guys will be working by yourself or with a partner to learn about how DNA, how those instructions in that blueprint actually then go on to code for proteins that go out to display your traits and do all the work in your body. Okay? So let me know if you have questions, and good luck.
I would. Can you put this on me?